I'm gonna start the lesson. Assalamu alaikum. Army Public School in College Khanewal. It's class second year and subject biology. It's chapter number 17, nervous coordination, and the topic is synapse and classification of neurotransmitters. Synapse. Yesterday we have discussed about the nerve impulse, the resting membrane potential, and the active membrane potential. Now we are going to discuss about the synapse. What is synapse? It is a consecutive neurons are so arranged that the axon endings of the neurons are connected to the dendrites of the next neuron, right? Here you can see this is one neuron and this is the second neuron, and you can see here the action potential is moving through the axon of this first neuron. Now it is moving towards the second cell neuron, right? What happened here? Here are the dendrite structure, the terminal end of this pre. This is the first one from which the message is going toward the next one. So this is the first one. It is known as the presynaptic cell or neuron, right? From here, the message is traveling, the nerve impulse is traveling. You can say the electrochemical message travels and reach towards the terminal end of this axon. Now it is attached with the postsynaptic cell that comes after. Okay, that comes postman after synaptic cell, and here this black area is showing the synapse, the area, the connection, the junction between these two presynaptic cell axon and postsynaptic cell axon is called the synapse. So the definition of synapse is cleared. A single neuron may form synapses with many incoming fiber of different neurons. So you can say that it can make connection with many neurons. Now next, let's see about the synapse types. We can say synapse can be the electrical or it can be the chemical. What is the electrical synapses? The electrical synapses and the chemical synapses. Okay. In electrical synapses, the nerve impulse moves very fast. Here you can see from this region, this is the presynaptic terminal and this is the postsynaptic terminal. And the electrical synapse that passes with the speed 0.2 nanometer in this axon. It is very fast. And the chemical synapse, it takes it speed is 20 nanometer. And how you can say that what is the chemical synapses? In chemical synapses, you can see that the direct nerve impulse doesn't move towards the postsynaptic terminal. Here it releases certain chemicals like these blue dots you can see here, right? These are the chemical, and the message that sent towards the postsynaptic terminal in the form of chemicals are called the chemical synapses. And the electrical synapses, the message that is sent in the form of electrical nerve is called the electrical synapses. It directly moves while it moves through the chemicals. See the next. Okay, before telling you the mechanism of synaptic transmission, let me tell you about few structure of the synapse, right? First of all, you need to know about, you are well familiar with the synapse, right? This is a junction. Here you can see again, this is the presynaptic, the terminal end of presynaptic axon, and this is the postsynaptic. Okay. Now you can see here the neurotransmitters. What are the neurotransmitters? Like the impulse, you can say the message that is moving in this axon, or you can say that is an impulse, take place in formation of a message which is transmitted across the synapse in the form of chemical messenger. Like the chemicals are sending message So the neurotransmitter which is transmitted across the synapse in the form of chemical messengers are called the neurotransmitter. So you can say that these red vesicles, or you can see them in this picture. Here you can see these vesicles containing the red color dotted areas. These are the neurotransmitters. So the neurotransmitters are the chemical messenger, right? These are the chemical messenger that will send message in the form of chemicals towards the postsynaptic cell. Okay. So the neurotransmitter is clear that these are the chemical messengers. Next is the synaptic node. Here you can see this knob-like structure, right? This end, the axon terminal end is knob-like, and it is called the it is called the synaptic node. Okay. Inside of synaptic node, you can see these are the vesicles. The red colors, the pink color vesicles are here. So these are known as the synaptic vesicles, which consist of neurotransmitters. Okay, like in this, in this image you can see. So these are the synaptic vesicles that containing these synaptic neurotransmitters. Okay, the neurotransmitters are the chemical messengers. So these three terms are clear to you, students. What are the neurotransmitter? What is the synaptic node and synaptic vesicles? Please let me know.
Yes, miss.
Okay. After that, we are going towards the mechanism of synaptic transmission, that how this chemical messenger moves from postsynaptic, presynaptic towards the postsynaptic. Okay, first one, the step number 1 that is given here. What happened in it? You can see that these are the calcium ions, right? This is the synaptic node. Okay, this is the area of synaptic node. Now what happened here? The action potential arrives at the synaptic node. In this node, the action potential comes. What is the meaning of action potential? You know, this is the area of axon. From axon, you remember action potential. The nerve impulse was coming in the form of action potential, right? Now it is coming here as the action potential is generated here at this synaptic node. What happened? You can see here, these are the calcium ions. Fine. Calcium ions open in the presynaptic membrane. You can see here, this is the presynaptic node, and the membrane which is present here is called the presynaptic membrane. Okay. Now the calcium will move inside of this presynaptic node. When the action potential is generated here, the calcium ion will come here. Okay. Now what happened? The calcium ions concentration inside the bulb is lower than the outside. So calcium rushes in. Fine. Now calciums are inside because first here was less calcium. Now the calcium has moved inside. The concentration is now high here. Okay. As the concentration is increased, synaptic vesicles, here are the synaptic vesicles. You can say this is a trigger. As the calcium ions come inside here, because of the production of action potential, they send these vesicles, they will send this vesicle towards the membrane. Here you can see that this was the vesicle, it is moving toward the brain. This is the presynaptic membrane. This is the first step, that voltage gated of calcium channel opens when the action potential is generated. Then the calciums are increased here and they send this synaptic vesicle towards the presynaptic membrane. The first step is cleared. Are you sleeping?
Yes.
Okay. After the first step, see the second step, the fusion. Now you can see the vesicle has been fused with these membranes. Okay. Who is speaking? This is the second step when these vesicles get fused with this presynaptic membrane. You can see this fusion, right? Neurotransmitter vesicles fuse with the plasma membrane of the transmitting cell. Now it has been fused here. See what happened next in the third step. You can see here it is the third step, fusion. Release the neurotransmitter molecules. Now you can see that here were the neurotransmitter molecules. They has been released in this space, and this space was known as the synaptic cleft, this area. Okay, now you can see that the neurotransmitter has been shifted into this synaptic cleft. After that, the released neurotransmitters, the released neurotransmitters, right? Molecules diffuse across the cleft. Now they will move across this cleft, fine, and bind to the receptor molecules. This is a postsynaptic membrane. Okay, here you can see these are the channels. Here they are going to be attached with them. Okay, so bind to the receptor molecules on the postsynaptic cell membrane. On the postsynaptic membrane, they are attached with the receptor molecules. Okay, what happened next here? From this diagram, you can see postsynaptic channels. They will let them to get inside. Okay, binding of neurotransmitter to the postsynaptic neurons receptors open some channels and allow sodium ions to diffuse across the postsynaptic membrane. From where we can see this picture, it's not given here. Okay, you know that students, here are the sodium and potassium ions are also present. As the sodium ions diffuse across the postsynaptic membrane depolarizes here. Do you remember the depolarization? After depolarization, when the action potential is generated here, then these neurotransmitters come inside of here. Now it has been reached here. It is you can say that this situation is known as the excitatory postsynaptic potential, because action potential is also generating here. Depolarization is also here. Sodium ions are also moving inside, right? And hence the neurotransmitters come and attached on these receptors, and it also moves inside. Now it needs to be, it is present here, right? It needs to be removed from here because we need to let in the other neurotransmitter. For this purpose, it has been hydrolyzed here with the help of certain enzymes. Enzymes are also present here. Like you can say acetylcholine is hydrolyzed by, if the molecule, let's suppose the neurotransmitter is acetylcholine, it will be hydrolyzed by the acetylcholine esterase and adrenaline by monoamine oxidase. So here are the two types of neurotransmitter that will be hydrolyzed by different enzymes. Now it will be removed from here. The next neurotransmitter can pass from here. So the action potential will move from with the help of the synapse, with the help of the chemical messenger, the neurotransmitter. Is it clear, student?
Yes, ma'am.
Good. Okay, now see them one by one. Okay. First thing what happened, action potential arrives at synaptic node, calcium channel open in presynaptic membrane. As the calcium ions concentration inside the bulb is lower than the outside calcium rush in, right? As the concentration increases in synaptic vesicle, move towards the brain. Not the brain. You can say the next cell. It can be the brain cell or any other cell. Neurotransmitter vesicles fuses with the plasma membrane of the transmitting cell. Did it happen so? You can see plasma membrane fusion. Fine. What happened after the fused vesicle released their neurotransmitter molecules into the synaptic cleft? You can see here they have been released into the synaptic cleft. What happened next? The released neurotransmitter molecules diffuse across the cleft and bind to the receptor molecules on the postsynaptic surface membrane. You can see here they has been attached on this postsynaptic surface membrane, these neurotransmitters. Okay. Binding of the neurotransmitter to the postsynaptic neuron receptors opens some channels and allows sodium ions. Of course, when sodium ions get in, you remember in action potential, the depolarization occurred there and action potential is generated again. Since the depolarization brings the membrane potential towards the threshold level, it is called the excitatory postsynaptic potential, and it is also known as EPSP. Next is, once the neurotransmitter have acted on the postsynaptic membrane, they are immediately broken down by the enzyme. Like they have reached on this postsynaptic membrane, they are present inside the channels. What happened? The channels need to be get closed, fine. And for this purpose, here are certain enzymes that will hydrolyze them. Like acetylcholine is one neurotransmitter that is hydrolyzed by acetylcholinesterase, okay? And adrenaline is another neurotransmitter that will be hydrolyzed by the monoamine oxidase. So these are the two types of enzymes which are useful for the process of hydrolyzing here. So the mechanism of synaptic transmission is clear to you all?
Yes, ma'am.
Okay, let's go and see the next one, the functional classification of neurotransmitters. We have classified them into two types. One is the excitatory neurotransmitter and the other is the inhibitory neurotransmitter. What are the excitatory neurotransmitters? Neurotransmitter that cause increased membrane permeability to sodium and thus trigger nerve impulse are said to be the excitatory. If you remember the depolarization, you must know the action potential, right? When it comes, what happened? In active membrane potential, the sodium ions come inside of the cell, right? And hence the action potential was generated there. This is known as the excitatory neurotransmitter. The neurotransmitter that were responsible there for the action potential are called the excitatory neurotransmitters. They are helping to send the chemical message towards the different neurons like glutamate. Next is the inhibitory neurotransmitters. Other neurotransmitter cause decreased membrane hyperpolarization, in which it refractory period, if you remember what happened there, can cause decreased membrane permeability to sodium ion, thus causing the threshold of stimulus to be raised. This section is called the inhibitory neurotransmitters. It lessens the chance that nerve impulse will be transferred to the adjoining neuron. Like the excitatory neurotransmitters are sending message towards the other neurons through the synapses, right? But inhibitory, like its name is showing, that it will not let them go. Fine. So it lessens the chance that nerve impulse will be transferred to the adjoining neuron. Its example is GABA and glycine. Amino acid gamma aminobutyric acid. Gamma aminobutyric acid and glycine. The endorphins are peptides. It is also example that function as both neurotransmitters and hormones, decreasing our perception of pain, that we cannot feel the pain. Why? Because that impulse which is responsible to send that perception is being inhibited by the inhibitory neurotransmitters. So the classification of neurotransmitter is also clear.
Yes, ma'am.
Yes, please. All right. It was all about our today's lecture. If you have any difficulty in any point, you may ask. Any question?
No.